First, I want to get started with a few housekeeping tips, as always, about tonight's format and also about the Q&A session. Tonight's format will be speaker presentation with a Q&A at the end. And so we encourage you to please send us your questions through the Q&A button, which you can find at the bottom of your screen. You can type those questions to us and we'll save them at the end for our speaker and also try to answer them along the way as well. Tonight's seminar will be recorded and it will be posted up on the UC Santa Barbara Natural Reserve System YouTube page. So after the seminar, you'll get a follow-up email through the email that you provided to register for this, and the link information to the seminar will be provided there. So you can watch it again or share it with your friends and family who didn't get to catch it tonight. So we've got another treat for you tonight. I'm very excited about this. Last week, we were at Sedgwick Reserve and we heard Professor Frank Davis tell us some exciting science and research about oak savannas. And we're gonna move a little bit west and south today back to Coal Oil Point Reserve. And I'm really excited for tonight's talk, both our speaker and also our intro to be provided by Dr. Cris Sandoval, our reserve director there. Before we get to our speakers, though, I just wanted to, share some background information with you about the University of California Natural Reserve System, which I might be biased, but I think it's one of the most amazing things that the University of California has to share. So what is the Natural Reserve System? It's essentially a network of protected lands in California that are set aside for the purposes of research, university level education, and also public service. The UC system altogether has 41 sites, and each UC campus manages a handful of them, and UC Santa Barbara manages 7. There's a lot of things that are really special about the reserves, and if you've been attending our seminar series so far, you've been able to get a glimpse of them. Like I said before, they're protected, unique ecosystems that are placed aside for research and for our students to learn. World-class research occurs at these places, and classes, instructors bring their students there as well. There's field station facilities that provide access to all these great ecosystems. And also there's resident and expert reserve directors who are there to help our researchers and help our students understand what it is that they're learning, they're using to do their work and to do their classwork. But most of all, what's important about the reserves are all the people that make it happen. And Coal Oil Point is probably the best case study of how community, university, friends, supporters, researchers all come together to basically create one of the best conservation and restoration stories I've probably ever heard, and probably all the students that are out there you've heard about in your environmental science classes too. So we're really in for some great information tonight. Coal Oil Point Reserve is the closest reserve to the UC Santa Barbara campus. And this is a great thing because it means that our UCSB students can walk there. And whenever I'm in town and I talk to people about the reserves, everybody always knows Coal Oil Point Reserve because people seem to have been there. There's a public beach. There's endangered species. We've got our western snowy plover there. There's federal critical habitat area, and also research and just all the things that come along with that kind of place. And it's all been able to happen, and everybody's been able to take a part of Coal Oil Point Reserve. And this is really due to the work of Dr. Cris Sandoval, our reserve director there. She's one of our longest serving reserve directors at UC Santa Barbara. She's been at the helm for over 20 years, and she's created, responded to, and to all kinds of environmental change, and has gotten us to the point where we're at, where I think we've got a really special place where research and teaching can happen. So Cris gets the wonderful honor of being able to introduce our speaker tonight. And with that, I'm going to turn it over to you, Cris. And again, thank you everybody for joining us. And please, Cris, take it away.
Thank you. Hang in there, everybody. Can you see my slides?
Looks good.
Okay. Well, welcome everybody. I'm really appreciate that you're able to get your eyes out of the vote count and join us here today. And I hope you can relax. I actually had to get a little help to relax and get myself out of the TV today. But yeah, so I manage Coal Point Reserve for now over 20 years. It's been just one of the best decisions that I could have in my life. Besides my family, it's just been an incredible journey. Myself with Jessica Newsom and Kip Callahan, which are full-time staff that help me out, forming a great team. And also Tony Nelson, who is our tea person part-time. We just have a blast here, doing everything we can to make this reserve better every day. As Marion said, Coal Point is very close to UCSB. We're just 4 miles west of UCSB. And this is a challenge and also great opportunities. And we see it that way, and we take those opportunities every time we can. And it's a small coastal reserve, but because we have so little coast in California that is protected, it's very special. And this land has a very complex history. You know, for 13,000 years, the Chumash lived here. And then in the late 1700s, the Spanish settlers removed the Chumash and brought to the mission system. And then in the 1800s, ranchers moved in and started creating pastures and agricultural fields. And you can see on this slide, the Devereux Slough in the middle, but pretty much everything around it, it has been denuded of vegetation. And then in the mid 1900s, 1940, oil was discovered in the coast of Goleta, and there's all these oil fields that were created, and the ranchers either leased or sold the area to refineries. So you would not believe it, but this area is just west of Elwood. Elwood is the place that today you go to look at this beautiful scenery and the sunset. It wasn't like that not too long ago, less than 100 years ago. And today, a lot of that ranching area have actually became towns, and it's full of houses, but Coal Point Reserve is still there. And really thanks to the vision of UCSB faculty that in 1970, they decided to protect this area. They realized that the reserve was this beautiful area full of native habitat that could be used by faculty and students to do their field work. So now this area is protected in perpetuity. And a lot of what we do is trying to get it back to what it was, because as you saw on that slide on the ranching days, the area was very impacted by those activities. Even though our reserve is small, it supports a large number of rare, threatened, and endangered species. Of course, our favorite snowy plover over there. We try to protect those species and recover them with the help of a lot of people. We have hundreds of volunteers, and we have particularly students or volunteers and help in our programs. We also provide over 50 paid internships to undergraduate students each year. So here you can see some students helping with the restoration and a docent there, a snowy plover docent that it's on the beach in charge of talking to people and tell them about the snowy plovers. The docents are there 7 days a week, all day light hours. It's just amazing. And we have students monitoring the Devereux Slough. Here is somebody counting tiny little invertebrates out of mud. And some students do their own research, and they actually, this is their first opportunity to do field research on their own, and that prepares them for grad school later on. We have also a lot of research projects going on, and because the reserve is open to the public, you might see those people out in the intertidal, and that's what they're doing. If you haven't been to Coal Point Reserve, I really encourage you to come. There are 4 miles of public trails that you can walk around and do a self-guided tour. And now it's my pleasure to introduce Andy MacDonald, who Dr. MacDonald I know for a long time because he does research not just at Coal Point Reserve, but also in a piece of property that I own on the other side of the mountain. And Dr. MacDonald is a disease ecologist who studies the ecology of vector-borne and zoonotic diseases. He's one of our own with a PhD in ecology from UCSB. He completed his postdoctoral training at Stanford University as a National Science Foundation postdoctoral research fellow in biology. That's a long name, Andy. And like many of us, he then returned to UCSB, where many of us are guilty of that, as a researcher at the Earth Research Institute. And Dr. MacDonald is particularly interested in how disturbances to natural ecosystems may affect our risk of being infected by parasites that would normally live in wildlife. And this is so timely right now, Andy, to have a talk about that. His talk today will focus on ticks in California, but he will also briefly tell us about his other research and adventures in Latin America and Borneo. And Andy, you can share your screen now.
Okay, how does that look? All right, so thanks Chris and Marion for the invitation to share some of my work this evening. And I appreciate all of you who decided to share your Thursday night with us here. So like Chris said, I've been conducting ecological research at Coal Oil Point now for quite some time, around a decade at this point. But my research has focused on a number of different diseases in a number of different regions around the world. And I'd like to just briefly touch on some of those before diving into focus on California, the UC Natural Reserve System, and Coal Oil Point. So some of my work has focused on mosquito-transmitted diseases in Latin America, including malaria in the Amazon basin and its relationship with deforestation. I've done work on climate effects on dengue transmission dynamics in Puerto Rico, and just had a major new initiative funded by the National Science Foundation in which we're looking at vector transmission across land use gradients in Latin America, particularly focusing on the Amazon basin. I've also done some work on various vector-borne disease systems in North America, including Lyme disease in the US Northeast and its relationship with forest fragmentation, as well as projected impacts of climate change on human Lyme disease transmission. And we have some new work that we're just getting underway on West Nile virus in California. Here we're looking at some impacts of agricultural land use change on the risk of West Nile virus transmission. Also have some work in the field of planetary health, which broadly is examining feedbacks between health and the environment. So for example, the effects of improved healthcare on forest conservation in Indonesia. And some National Center for Ecological Analysis and Synthesis supported work. So this is a center based here in Santa Barbara that's focused on identifying ecological levers for health. So in other words, ways in which we might intervene to simultaneously achieve human health and conservation objectives. And even got pulled into the fray a little bit to do some work on COVID-19 as well. So I won't have time to talk about all of that today, but I wanted to bring it up in case any of these topics of research interest you and you want to learn more. I'll put my contact information on the last slide, so feel free to reach out if you want to talk more about any of that. So today I'll be focusing instead on my work on the ecology of ticks and tick-borne disease in California, which has made really heavy use of the UC Natural Reserve System over the past 10 years or so. I would say without the UC NRS, my research in this area would not be feasible, and I wouldn't have been able to complete the PhD that I completed. Of the dozen or so reserves that I've sampled or conducted studies in, Perhaps most notable is the extent of my use of UCSB's Collier Point Reserve, which has played a role in many of my published studies, and it continues to be, I think, one of the most interesting sites that I work, which I think you'll see why shortly, as I discuss tick-borne disease in a changing California. So I'd like to start by giving you a brief history of my connection with Collier Point and how it's played a role in shaping my science and my interest in the natural world. So a little over 10 years ago, I moved into the West Campus apartments here adjacent to Dever Lagoon and Collier Point. Initially, I thought this was great because I could go up to my garden plot and have a view of the slough, even the ocean on a clear day. I could go on a walk or a run around the slough and along the adjacent Elwood Bluffs. I could walk to the point for sunset, as many do, and there have been many a sunset over the years. It offered great opportunities for things like beachcombing and tide pooling. And I actually had the opportunity to teach undergraduates at UCSB about marine intertidal invertebrates at Collier Point for a number of years in a row as part of an ecology course offered here. Now, eventually in 2012, after what felt like the requisite ill-fated project or two at the beginning of many of our PhD careers, I decided that I was going to study the ecology of ticks and tick-borne disease, and that I was going to use the UC Natural Reserve System and Collier Point to do so. And I thought, how cool is it that I can walk out my front door and be doing research in a matter of minutes? So here are just a few shots from some of that research. So here I'm checking a mouse for attached ticks that might be feeding on it and taking a tissue sample from it to screen for infection with the bacteria that causes Lyme disease. I trapped it using this live trap, which was baited not far from the pond trail that winds through the reserve, for those familiar with it. On the bottom, on the left, I'm setting out some temperature loggers to track abiotic conditions in some of the plots that I've sampled over the years. And then in the bottom right, I'm checking what we call a drag cloth in my line of work. Essentially, it's just a white piece of flannel, but this is how we collect ticks in a standardized way. So if you are walking through the reserve and you see someone with one of those, that's probably what they're doing. And I should note that I've also been lucky enough to teach undergraduates these exact sampling methods at Collier Point as part of a field methods course that's offered through Santa Barbara City College. So the instructor for that course has reached out to myself and other UCSB scientists over the years to lead lab exercises for the class, which I've done at Collier Point. Now, in addition to being an excellent place to live, an excellent place to recreate, a great place to teach students about the natural world with its proximity to UCSB's campus, as well as a great field site for conducting ecological research, Collier Point is also a very dynamic place. So some of that dynamism is natural with the ever-changing coastline, intertidal habitats, the slough which periodically breaks and refills. So this is a shot of the slough emptying out into the ocean from just a few years back. I think Cris actually was out there with me and we got to watch this event unfold, which was a pretty cool thing to be able to see. But some of that dynamism is increasingly coming from processes of global change. So from droughts and wildfires that are influenced by climate change to increased human pressure associated with population growth and conversion of natural lands for development. So this is a shot taken from the edge of Devereux Slough of the Sherpa fire in 2016, spewing smoke and ash over the coast. And it's the impact of these processes of global change on vector-borne disease transmission and human health that most interest me and that I'll be discussing in this talk. So for example, ticks and mosquitoes, things that transmit pathogens to us, they're ectotherms, so they're sensitive to temperature. So as temperatures change across the globe, this is likely to shift the ranges of these organisms and the regions that are most likely to be impacted by the diseases that they transmit. In addition to climate, vector species and the hosts that maintain pathogens in natural transmission cycles are also sensitive to habitat loss and land use change. So different host species will respond differently to habitat loss due to things like differences in body size or resource requirements. So land use change will result in changes in the composition of host communities, so what species are there, which could influence the abundance and rates of infection in vector populations. So for the transmission of vector-borne diseases to occur, it requires a number of different conditions are met. And you can sort of think about these as a series of filters that must align. So for example, host species and vectors need to be present and abundant. Vectors must come into contact with the right host species. Vectors must be what we call competent, which essentially means capable of acquiring infection and then transmitting new infections to new hosts. And hosts must also be susceptible to the pathogen. And of course, all of these different processes and interactions are influenced by the environment and environmental change. So things like habitat influence vector and host distributions. Temperature and humidity might influence vector competence or vector host contact rates, and so on. So it really requires an ecological lens to understand the environmental drivers of vector-borne disease transmission and to make predictions about what we think will happen in the future. So for the rest of this seminar, I'm going to discuss some of the mechanisms by which environmental change influences tick-borne disease ecology and transmission in California. And I'll start with some of my work on the influence of climate and climate change on the distribution and activity patterns of ticks. I'll follow this with a brief discussion of the effects of local environmental and abiotic conditions on ticks and the pathogens that they vector. And we'll start getting into the importance of hosts and host communities in this section. And we'll explore the effects of wildfire disturbance on the community ecology of tick-borne disease. With the fire seasons that we've had lately, I think this is probably a timely thing to discuss. And to wrap up, I'll just discuss how we might integrate what we know about the ecology of ticks and their hosts to predict regions of high transmission risk for Lyme disease, both now and under projected climate change. So let's start with the basic ecology of this system. So the pathogen responsible for this disease is this spirochete bacteria called Borrelia burgdorferi. In California, it's transmitted to humans by the tick Ixodes pacificus, or the Western black-legged tick. And this tick has a 3-stage life cycle. So the larval stage hatches from eggs, which emerge to feed in the spring. Each of these life stages feeds only once per year, and the life cycle takes 3 years to complete. Importantly, for Lyme disease, larvae hatch uninfected. So infection must be acquired through feeding on an infected host. There are a diversity of other tick species in California, some of which you may have encountered at Colloidal Point as well. And some of them may actually play an important role in the ecology of this bacteria, which we'll talk about later in the talk. Finally, different host species play different roles in the ecology of both the tick and the bacteria. So for example, wood rats and gray squirrels are important reservoir hosts for the bacteria. So what we would call a competent host. And lizards, on the other hand, particularly Western fence lizards, the ones that you see doing push-ups all over the place, these are what we might call non-competent hosts. These lizards actually have a protein in their blood that kills the bacteria, and they also cleanse feeding ticks of infection. So if an infected tick feeds on a lizard, it will drop off of that lizard uninfected. This is great, but there's a caveat. Lizards also feed a really large proportion of Western black-legged juvenile ticks. So in a sense, they also act as a tick population amplifying host. So if you remove lizards from the system, you actually see population declines in this tick species. And large mammals, namely deer, are important reproductive hosts for adult ticks. Adult ticks that feed on deer will produce large numbers of eggs, up to multiple thousands, and are thus important for maintaining populations of this tick species. So you need a lot of these elements of the ecology of the system to align in order for what we'd call spillover transmission of this bacteria to humans to occur. In California, ticks infected with the Lyme disease spirochete have been identified throughout the state. So in every county on this map, with the exception of those in gray, where ticks have been collected but not found to be infected, or those in white, where no ticks of this species have been collected. But transmission of this pathogen to humans is not equally likely across this range, and human disease incidence tends to be much higher in northwestern California than in central or southern California. So here, any county in gray has reported human cases, but incidence is quite low, while those counties in white have no cases reported. So to explore the ecology of Lyme disease in California, I'm going to try and address a number of these filters using studies that we've conducted in the field, in the lab, as well as behind computer screens. And I'll start with the geographic distribution and seasonality of host seeking of this tick species. So again, as small ectothermic organisms, ticks are really strongly influenced by abiotic conditions like temperature or precipitation. Vegetation cover is also key because it determines both microclimatic conditions that ticks actually experience, and it provides the structure from which ticks seek hosts. So each of the colors in this map represents a distinct vegetation type. We can leverage occurrence records for this tick species, along with these remotely sensed climate and vegetation data that I just showed you, to predict the distribution of the vector. So here, each of these points represents a location where individual ticks of this species have been collected by the California Department of Public Health during routine surveillance that they conduct throughout the year. Using a species distribution modeling approach that essentially compares the distribution of environmental or climate characteristics at locations where this tick has been collected to a set of randomly selected background locations from across the state, we can predict the suitability of environmental conditions for this tick species. So in this figure, I'm showing you the result of a model that uses annual average climate conditions to predict essentially the overall distribution of the tick. So in this case, red indicates high suitability for the Lyme disease vector, while blue represents very low suitability. So anywhere in red, we would expect to find this tick. As you can see, suitability is high along the coast and in coastal mountain ranges, particularly in North Coastal California, as well as in the Western Sierra Nevada foothills. But we were also keen to adapt this approach to predict seasonal activity. So in other words, when is the tick actively seeking hosts for a blood meal? And this is really important ecologically for things like pathogen transmission dynamics, but it's of course also a key determinant of human risk of tick encounter or tick bite. So what time of year are you likely to encounter this species? So our expectation is that seasonal activity patterns will be limited by adverse abiotic conditions, like for example, higher temperatures or low humidity. And this is just a time series of environmental conditions in a few natural reserve system sites that I've worked. So you can see that Coyle Point in blue tends to be more humid and has cooler summers than Sedgwick, for example, in red. And from a series of field studies that I conducted throughout the UCNRS, we can see that as you move along California's latitudinal gradient from north to south, so as we go down each of these panels, we see that tick abundance declines. So in this figure, from around 20 to less than 2 nymphal ticks per 100 square meters. And the period of seasonal activity also declines from over half the year in the north to only 3 to 4 months out of the year in the south. So in each of these panels, we're representing tick density plotted through time. So each dot represents a sampling event and the gray curves represent tick abundance. So the width and the magnitude of those curves shrink as you move south. But these field studies are somewhat limited in geographic scope. So it's really challenging and takes a lot of effort to replicate that sort of effort across large and heterogeneous geographies. So we decided to leverage the same tick collection database to help fill in the gaps. So we subset the observations by month, and instead of assigning the annual average values of our environmental predictors at each location, we now have the conditions in the month of collection at that location. So that'll help us better approximate conditions that were actually experienced by the ticks when they were observed in the field. We then can project this model onto monthly climate data to predict when ticks will be actively seeking hosts in each month of the year. So these are analogous maps to one that I just showed you with high suitability in red and low in blue, but now for each month of the year. So these monthly results indicate that host-seeking activity is high across the distribution of this vector species from around December to April. But if we look at regions of South Coastal California and to some extent the Southern Sierra Nevada foothills, the probability that ticks will be out and active is low by May, as well as in the early season in November, in contrast to Northern California, where they're still predicted to be active during these times. So this suggests that ticks are active for a larger proportion of the year in Northern California, meaning a more protracted risk of tick encounter in this region, as well as a longer window for host feeding. So this might influence things like tick abundance and pathogen transmission. From this, we can also gain some insights into what might be influencing host-seeking activity. And in this case, we find that monthly precipitation and maximum monthly temperature are some of the most important predictors of seasonal activity. So these are response curves that indicate the relationship between the probability of tick activity and each of the variables. So for example, the likelihood that ticks are active increases as monthly precipitation increases, but asymptotes quickly. And maximum monthly temperature shows this interesting unimodal relationship, where likelihood declines at both high and low temperatures. So this could be, for example, the key factor that limits activity earlier in the season in Southern than in Northern California as summer temperatures increase. Vegetation was also a really important element of this model, and the most suitable vegetation types were hardwood forests and oak woodlands. So if you're going for a hike in winter through an oak woodland, you might want to watch out for these ticks. So we've also modeled the distribution and seasonal patterns of activity under projected climate and land cover change. So here I'm just showing annual predictions for, on the top, a hotter and drier climate change scenario with the change from current illustrated on the right. So those rightmost maps, in those cases, red indicates an increase and blue a decrease in suitability. And then on the bottom, we have a not as hot and wetter scenario. So you can see that suitability increases in the North Coastal and Sierra Nevada regions, while decreasing in the South under the hotter and drier scenario, and actually increases slightly across the state in the wetter future scenario. So there's a lot of additional detail that we could dive into here from this study, but I'll save most of that for anyone who has a particular interest. Suffice it to say that our predictions vary quite a bit regionally and from one climate projection to another. So here, for example, we have plots of seasonal suitability for the northern, central, and southern California coast regions. So in all of these cases, suitability is high in the winter months and declines into the summer months. But in the northern region, so that figure on the left, we largely expect increases in suitability and slight increases in the amount of time that ticks are out seeking hosts under the hotter and drier scenarios in red. So if you look at those red scenarios, they are the curves are above the gray, which is the current. So in this case, in the winter months, so you can see that those red curves are higher than the gray curve in the winter months, and they decline somewhat more slowly in the summer months as well, which indicates longer host-seeking seasons. The opposite, though, is true for the wetter scenarios. So those are mapped out in blue, and in this case, we expect seasonal activity to decline substantially. In the central coast here where we are, we don't expect too much change, but in the southern coast region, we see actually the opposite predictions from the northern coast, where suitability is expected to decline in the hotter and drier scenarios, but increase in the wetter scenarios. So in addition to projected directional shifts in average annual or seasonal climate conditions, climate change is also likely to lead to more extreme weather events like droughts. So this is a time series of the Palmer Drought Severity Index summarized by month in each of the 14 different UC reserves that I've collected ticks from over the years. So a handful of these, including Coal Oil Point, I've been consistently sampling every year since 2013. So in this figure, any value below 0 indicates drier than usual conditions. And usually this metric doesn't exceed -4. So anything below -4 could be considered extreme drought conditions. So as you can see from the time that sampling began in 2013 through about the end of 2016, all of these reserves were experiencing pretty severe drought conditions until it finally broke in 2017. So this long-term observational study, again, of which Collier Point is a part, began in the winter of 2013. And I've gotten substantial help from reserve staff and managers across the UCNRS. And we've been compiling a time series of tick abundance. So this figure is just a snapshot in time from 2014 with the size of the circles scaled by tick density. Each color represents a different tick species. And in this case, red is the Lyme disease vector. So this sampling, of course, has spanned the recent drought as well as California's emergence from it. So along with UCSB ecology PhD student Samantha Sambato, we're leveraging this natural experiment to understand the impact of drought on tick populations and infection with tick-borne pathogens. And some very preliminary results from just a small subset of the data that we've been able to process so far. So we have lots of boxes full of ticks from across the UCNRS in my lab right now that we're still working on. It appears that drought, when we control for other climate conditions, may lead to reductions in tick abundance. So in this figure, as you move to the right on the x-axis, drought conditions become less severe and tick abundance increases. But the relationship, at least so far, is pretty noisy. And we hope to resolve this with more data as we continue with tick identification from collections across the reserves, as well as start exploring some of the impacts on infection with tick-borne pathogens. So we've learned a great deal about where this vector species is distributed, its host-seeking activity, and how these might change under projected environmental change through both field observations across the UCNRS, as well as through these other data-driven studies. But to learn where this pathogen is and what species are maintaining it in nature, particularly locally here in Santa Barbara, it really requires local and targeted field studies that the UCNRS supports. And it's what we've learned from some of these studies at Collier Point and some other UCSB NRS reserves that I'll discuss next. So while the studies that I just shared are useful to give us a sense of large-scale distributional or activity patterns and how those might change as the environment changes, the reality is that ticks are also responding to really micro-scale variation in climate and habitat. So to understand local patterns of tick abundance and infection prevalence of ticks in Santa Barbara County, we established a series of plots distributed throughout Collier Point, Sedgwick, and Paradise reserves, in which we measured things like microclimate, physical properties of the sites. We estimated host abundance, quantified habitat types and vegetation cover, and so on. And from this sampling effort, we identified this interesting shift from host abundance being the key factor influencing tick abundance early in the study. So this is starting in 2013. To microclimate and habitat conditions really driving tick abundance, particularly for the small-bodied juvenile stages. So the backdrop of this, of course, is the progression of California's historic drought. And infection prevalence in ticks was primarily influenced by temperature and the diversity of the tick community. So namely, higher diversity tick communities tended to have higher rates of infection, and higher rates of infection were found in plots with low seasonal variation in temperature. So we probed this question of tick diversity a bit more, and it turns out that some of these species may also be competent vectors of Borrelia. So for example, the rodent specialist species Ixodes spinipalpus. And here in this figure, we're summarizing infection across all these sampled sites in tick and small mammal species, with Sedgwick on the top, Paradise in the middle, and Collier Point on the bottom. So we found here that infection is more common in rodent specialist tick species like Ixodes spinipalpus, and this other species that we found at Collier Point called Ixodes paramiski, much more common than it is in the human vector, Ixodes pacificus. And these species appear to maintain higher rates of infection in small mammal host communities as well. Interestingly, Ixodes paramiski, at least to my knowledge, has only previously been collected on the Channel Islands. So this finding in itself might suggest some interesting connection between the islands and the mainland at this location that I'm excited to explore moving forward. We find that these other possible vector ticks are much more strongly associated with abiotic conditions and habitat conditions near the coast at Coal Oil Point. So specifically, they exhibit a really strong negative association with maximum average summer temperature. So even within Coal Oil Point, they tend to be found in microhabitats that experience very limited temperature extremes. And we found a sort of remarkable diversity of strains of Borrelia that were in circulation in this region. So Borrelia burgdorferi sensi stricto, or SS, in yellow at Coal Oil Point, is the strain that's most commonly associated with Lyme disease in North America. But Borrelia bezetiae in blue, which is the most common strain that we found locally, is actually thought to be involved in Lyme borreliosis in Europe. And as you can see, infection in both small mammals and ticks is much more common at Coal Oil Point than the other sampled reserves. So this all suggests that this pathogen is being maintained locally in natural transmission cycles that involve small mammals and specialist tick species, but that spillover transmission to humans is likely rare due to a relative dearth of infection in the human vector. So you can see that of over 600 or so Western black-legged ticks labeled I. pacificus here that we've screened so far across these reserves, only 2 have tested positive. So finally, given the recent extreme fire seasons that we've experienced here in California, I'd like to discuss the effects of wildfire on the community ecology of Lyme disease in California. And I'll use the White Fire, which occurred in 2013 off Paradise Road north of the Santa Ynez Mountains, as a natural experiment to explore this. So here we set out to explore the response of the whole community of species involved in the ecology of Lyme disease to this disturbance. So we tracked the response of ticks of all the different life stages. We tracked small mammal populations and communities using live trapping techniques. We measured non-competent lizard hosts using news captures and site recite methods. We tracked activity of deer, which again are reproductive tick hosts, using scat counts, as well as tracked Borrelia in ticks and in hosts, both within and adjacent to the burn extent. And what we found here suggests a nonlinear response of tick-borne disease risk to wildfire disturbance in California. So there was elevated risk within the burn extent in the winter and spring immediately following the summer wildfire. So what we saw was elevated abundance of adult and nymphal ticks on the landscape. And I think this was due to reductions in host populations for these ticks that resulted in elevated abundance of questing ticks. So essentially what's happening is these ticks survive the fire and they're out looking for hosts that aren't there in the same numbers that they used to be. And we also found evidence for infection in ticks and in hosts within the burn sites. So in this plot, red arrows indicate significant positive and negative effects of wildfire, so up and down respectively. And we see significantly elevated abundance of adult and nymphal ticks in the top and middle figures. And this is in burn sites in white, unburned sites in gray. So that horizontal line in these figures represents the average tick density across the sampled plots. So you can see it's higher within the burn sites for both nymphs and adults. Yet if we go forward in time to look at the effects 2 and 3 years on, risk declines substantially in subsequent years, with evidence suggesting that long-term risk reduction through effects on tick populations and key host species occurred. So we observed reduced abundance of all tick life stages within the burn extent in 2015 in the middle and 2016 on the right. So you can actually see that no juvenile ticks were collected in either year within the burn extent. These reductions may have been due to a couple of different factors. So one, potentially reduced feeding success resulting from lower host abundance, or potentially to mortality associated with changes in habitat and abiotic conditions that resulted from the fire. So for example, we threw some temperature loggers out there and they suggest that temperatures are much higher in the burn sites than in the adjacent unburned sites. So that have led to desiccation of ticks and higher rates of mortality. There was also a period of significantly reduced activity of deer in the burn sites. This was a bit surprising to me, though activity rebounded to similar levels to the adjacent unburned sites by the third year of the study, illustrated in the rightmost panel. So you can see there that average deer activity looks about the same in the burned and the unburned plots. We found that fire destroyed wood rat nests and no wood rats were trapped within the burn extent. So again, wood rats are an important pathogen reservoir in the system, but there was no clear effect of fire on fence lizards or mice. So taken together, this suggests that wildfire initially amplified risk through elevated abundance of host-seeking ticks in the year following the fire, but that loss of key host species and declines in deer activity and tick populations within the fire perimeter may have served to reduce risk in the longer term. So from these studies, we've learned a lot about the importance of the ecology of the system to understanding the distribution, abundance, and infection of vector ticks. But what we'd really like to do is get here. So understand how this ecology influences human disease transmission. And this is a much harder thing to wrap your head around. So some of our ongoing work is taking what we know about the distribution and habitat suitability for key host species, the distribution of the tick vector from those earlier studies, where humans are likely to come into contact with the vector tick, where infected ticks have been collected to predict where vector competence and host susceptibility are high. And if we integrate all of these key elements of the ecology of this disease from these local and targeted field studies, along with larger scale data-driven approaches, can we accurately predict where Lyme disease transmission to humans is likely to occur, both now and under projected global change? So that's where we're moving next. With that, I'd like to acknowledge many collaborators on the work that I presented today. And specifically, I'd like to acknowledge the support of the UCNRS. So I've received monetary support through a number of Matthias grants and support from ICIKI when I was a graduate student. And of course, the UCNRS provides the living laboratory that made much of this work possible. And I'd specifically like to thank Cris and her husband, Kevin, who you may have heard from earlier in the series, for their support. Both at Coal Hill Point and Paradise Reserves. That's been huge, and I can't thank them enough for that. So with that, I would be happy to take any questions.
Great. Okay. Thanks, Andy. If you want to pull down your slides, we've got a bunch of great questions out there, and thank you for a really exciting talk. I did not know all of that, so I've learned a lot tonight, and so I'm just going to go ahead and get started. We've got a first question from Joelle Canto-Adams, and she asks, in your models for tick density and transmission abundance, how big of an influence is human population and infrastructure on density? And she's asking, would this explain part of the difference between Northern California and Southern California?
Yeah, it's a good question. So in those models, we didn't include human density per se. I suppose you're maybe thinking about human habitation and density, sort of removing habitat for ticks and their hosts. We do include vegetation in those models. And so it's true that there is probably just less suitable habitat in Southern California, in part as a result of there being so many more people distributed on the landscape. So yeah, that's a good question.
Okay, Alex Sims asks, can we get Lyme disease from those rodent specialist ticks, not the main tick species you were discussing?
Yeah, I thought that question might be asked. I don't want to say that it's impossible, because it's not impossible, but it's extremely unlikely. So that's for a couple of different reasons. One is that because they're specialists on particular host species, they're seeking for those hosts in really different ways from the human vector. So the human vector, you might brush up against a blade of grass that it's up waiting for some host to walk by. These species, they tend to be searching for hosts down in the leaf litter or in places where people just aren't likely to come into contact with them. And some of these species, because of the hosts that they've evolved to feed on, are actually not capable of biting humans. So they might be able to break through the skin of the ear of a mouse or something like that, but not human skin. So even if they found themselves on you, it would be unlikely they could actually bite you.
So it pays to have thick skin, everybody out there. So
lots of ways, yes.
All right, so here is a question from Scott Cooper, and he's asking, in projecting the effects of climate change on tick abundance, you used climate tick distribution models. If you refined your model to look at climate change effects on the variety of hosts, would that change your predictions? Yeah,
that's a really good question. That was something that we couldn't easily incorporate into the model that we ran. Sort of suggesting that tick hosts are likely to also change their distribution under projected climate change. We suspect that a lot of that will be captured in changes in vegetation type, which we do include in the model. So ideally, we're capturing some of that, but we're not including changes in host distribution directly to model those processes.
Thank you. So here's a question that was on my mind too. This is from Dolly and Charles Mullen. And they're saying, it seems like an odd thing to ask, but do ticks have any kind of potential positive ecosystem effect? Would we miss them if they were eradicated? What animals feed on them?
Almost no animals feed on them. There was an attempt a while back to use guinea hens in the Northeast as sort of a biocontrol for ticks. And as far as I understand, it really didn't work. Ticks are pretty bad eating unless they've just taken a blood meal and they're full of blood. And at that point, they're basically going down into the leaf litter and they're sort of hard to access. So my feeling is that it's unlikely that they would have a really large, sort of negative impact if they all of a sudden disappeared. It's hard to say for sure, but there just isn't that much out there that relies on them as a resource. So they're really just out there being parasites.
They're out there being parasites. All right. So here's a question from a local expert, Kevin Lafferty, and he's asking, he's saying Coyle Point has a ton of rabbits, but no deer. Is that why Lyme is common at Coyle Point, but risk to people is low?
A ton of rabbits, but no deer. So that is a good observation that there are not deer at Coyle Point, although I did on one occasion see a fawn, a dead fawn on the side of the road by the slough. I don't know how it got there, but it was there. I have photos if you want to see them. It's true that you don't find that many black-legged ticks at Coyle Point. I suspect that the ones you do find might actually be transported in by birds or something like that. And there may not be an actual self-sustaining population there. So it really is all about these other tick species that, again, are not likely to bite a human. So we've got circulation of the pathogen, but it's really unlikely to make that jump into humans.
So here's a question from Cesar Carrasco, and he's asking, what internships would you recommend for students interested in studying virology? And what path led you to studying vector-borne disease?
Oh, internships. I mean, I would say contact any of your local disease ecologists and see if they might have some openings in their labs. Myself and others that study infectious disease at UCSB and elsewhere, we're always looking for help. So I would say, contact folks at your local institution. As far as how I got into this, it was kind of an accident, I suppose. So I mentioned that there were a few ill-fated projects at the beginning of my PhD. I set out to study the dynamics of an agricultural pest, actually, that was just invading California at the time. That project didn't make it very far because they were pretty afraid of having anyone working on this thing at the time. It was the Asian citrus psyllid. And so I was out in the field trying to get some sort of project set up. I returned to my office to find that I had a tick on me. And that sort of planted the seed, like, oh, maybe this is something that I could study for my PhD, and sort of all history from there.
That's great. So it literally just bit you. Great. Here's a question from Maya Chen, and Andy, you can answer this, and maybe Cris, you might want to respond to this too. But great question. What kind of effect does the ecological restoration of Coal Oil Point Reserve and other sites have on the abundance of certain hosts and tick populations? This is a two-part question. Are there any predictions of how the combination of restoration and possible climate change patterns will interact to impact the abundance of host and tick populations?
So the places at Coal Oil Point that I tend to find these ticks that have been infected with Borrelia, it tends to be in the leaf litter under the coast live oak canopies. And so the extent to which we're bringing coast live oak back, that might increase habitat for ticks and their hosts. It's hard to say for sure, but I don't think that's a good reason to not restore those habitats. They have lots of other important services that they provide that I think outweigh any potential negatives that could happen as a result of doing that.
Great. Okay. Here's kind of a fun one. Elise Cypher asks, I found a tick on my hand once at Coal Oil Point Reserve. Do you think it would have bit her?
If it wasn't stuck to you, then probably not. So ticks, it takes them actually a really long time to take a blood meal. Once they find a place that they're okay with attaching, which can sometimes take 24 hours, they usually take around a week to feed. It sort of depends on the life stage. But if it bit you, you would have noticed it. It would have been stuck to your hand, engorged. If it was just walking around, there's really no chance that it bit you.
Yeah, see, that thick skin comes through again, comes through again, everybody. Well, we're just about out of time. I'm sorry that we could not get to everybody's questions tonight, but we had a lot of good questions and great talk, Andy. I really appreciate that. And I encourage everybody to join us, please, next week. We're actually going to be moving to the other side of the mountains, and we're going to head up to the Eastern Sierra, where we're going to hear a talk from Dr. Tom Smith titled Think Globally, Act Locally, How a Pandemic Challenges Conservation of a California Frog and How Scientists and Managers Can Respond. And that's hosted by the Sierra Nevada Aquatic Research Laboratory, one of our 2 reserves that are located up in the Mammoth area. So please join us for that. And once again, I just want to say thank you to everybody for joining us. Thank you, Cris, for sharing about Coal Oil Point Reserve with us. And again, thank you, Dr. Andy MacDonald, for this fascinating talk. I hope everybody has a lovely evening, and hope to see you next week as well. Thank you.